Precision medicine to pulmonary vascular disease is not new, and several of the other speakers have already highlighted this. But one of the things that has come up is that in order to do this and do this right, that we need to actually include all of the genomics that have become critically important to pulmonary vascular disease, along with clinical phenotyping and deep clinical phenotyping, as well as incorporate omics as well, to be able to create these endophenotypes that will help us understand the disease. Now, you saw this earlier today, and one of the facts and features of precision medicine is that you can actually have individuals with different endophenotypes that once you try to look at them more closely with clinical phenotyping and information, that you can find out more about them. But most importantly, what is also necessary is what we call panomics profiling. And this is really just refers to this piece right here, which goes beyond metabolomics and beyond proteomics to include such things like exposures and creating exposomes, looking at microbiomes, and all sorts of other aspects that need to be incorporated together and, as just brought up, need to be analyzed in new and creative and innovative ways to integrate the data to be able to understand what the phenotypes and the phenotype clusters are. Now, the NIH had been talking about this for a while, and this was really what gave birth to the idea of PVD-omics, or the Pulmonary Vascular Disease Phenomics program. And the idea was that the study would look at everything from molecules to man and try to integrate and incorporate all of these things together to get a large cohort in the US of very intricately and deeply phenotyped individuals to try to understand, is our current classification system of patients adequate? Can it be improved upon by using omics and other phenotyping to find subcategories and subclusters of individuals? Or is it really that we're just using all of these things to risk stratify patients? For those that are not familiar with the PVD-omics setup, it is a large multi-center study that is governed by a steering committee and a data coordinating center. There are several clinical sites around the US, and there are multiple cores that are looking at both the clinical phenotyping data as well as all of the omics phenotyping data. And as of right now, the planned omics include coagulomics, cell biomics, metabolomics, proteomics, genomics, and again, even more expansive profiling than that. Now, one of the first things I want to point out is that the clinical phenotyping of the patients is extensive. And so this is typically spread out over several visits, and it all takes place within 6 weeks of each other. In addition to doing things like getting your typical extensive history physical exam, we are also getting exposure histories, reproductive histories, childhood histories. We are getting blood specimens that are being done at the initial visit for routine labs and non-routine labs. And then all of the patients undergo pulmonary function testing, echocardiograms with specific focus on the right ventricle, cardiac MRIs to add more information, sleep studies, 6-minute walks, and importantly, they get right heart catheterizations with challenges, and they undergo cardiopulmonary exercise testing. In some of the centers, including ours, they are also undergoing invasive cardiopulmonary exercise testing, and at each of these different visits, individuals are getting biospecimens taken both in the form of blood, urine, and we're taking them from multiple different sites. So we have peripheral, we have pulmonary artery, we have arterial, and we actually have them at different time points from the invasive cardiopulmonary exercise testing, where we have paired samples so we can look at things like transpulmonary gradients. As Roham just brought up, The idea of having a shared protocol is terrific. If anyone is interested, the PVD-Omics protocol has been published, and there are links to the data collection methods, as well as the blood specimen collection and processing methods, et cetera. So that and the entire rest of the protocol is out there and available. Where are we right now? Well, yeah, at this point in the study, this is actually old data because this is from the beginning of May. We are now past 600 people enrolled. And as part of this study, we are enrolling control subjects, so healthy controls with no evidence of cardiopulmonary disease. We are enrolling disease comparators for each of the WHO groups, and that we have both subcategories of mild disease comparators and moderate disease comparators, and then individuals with group 1 to 5 PH across the board. And as you can see, about 75% of individuals enrolled have pulmonary hypertension, and then we have 18% as comparators and healthy controls. The overall goal is 1,500 total, 1,000 with PH, 400 disease comparators, and 100 healthy controls. Now, within this study, each of the groups has been looking at some different sub-studies that they are doing, and I'm showing here some work from Evan Britton's group at Vanderbilt, where they were asking the question, do individuals with combined pulmonary hypertension behave more like pulmonary arterial hypertension or isolated post-capillary pulmonary hypertension? And interestingly enough, when they did genetic screening and identified gene ontology groups, they found that there were a number of factors where individuals with CPC looked more like PAH than group 2 PH. And I will just point out for Nick, HLA, DPA1, and DPB1 came out in the profile, which Evan pointed out as you were speaking. So it's actually quite good to see that there's correlation between some of the different studies, or the same hits are starting to come up. Other groups from the study have been doing proteomic screens. So proteomics has been done using pulmonary artery endothelial cells that have been collected from patients, looking at PAH versus control. Using mass spec, they identified over 110 differentially expressed proteins from the cells, and the top 5 actually are listed here. At our center, we are interested in exercise, and because we are collecting blood from both the pulmonary artery and the arterial blood at the same time, we are actually using exercise as a perturbation to try to unmask dysfunction. And we did this and then did a proteomic screen. And what you can see is in healthy control, well, let me say controls, they're not healthy because they're having this test. But these are people who did not have PH. And on the basis of their transpulmonary gradient, when you compared the way they were at rest and post-exercise, we found 41 differentially expressed proteins that map to pathways, including immune system activation. When we do the same analysis, looking at individuals with PAH, what we see is now a different set of genes that are differentially expressed by exercise, and these actually map to some different pathways, some of which, such as regulation of STAT3, we thought was encouraging because this actually fits with the disease pathobiology, and then a number of other things that we saw that were also interesting. Metabolomic screens have also been done, again, looking at just a number of PAH patients and controls, showing some differences in biochemicals and principal component analysis, shows that both of these can be used to differentiate between PAH and control patients. And in addition, some other interesting things looking at coagulomics. So one of the things that we don't actually talk about so much, showing that D-dimer expression levels in individuals with exercise-induced PH were actually different than what was seen in control subjects. And if you looked at the transpulmonary gradient of D-dimer, that this actually correlated well with the VO2 max, or a marker that's used to understand patients' exercise capacity. So what are we doing with all this, since all of those were just some individual studies with few of the patients that are in the cohort? We now are going to have a 1,500 patient cohort of PAH and comparators, which will have a large variety of biological samples that can be used for this pan-omics profiling, and that will do the sort of typical omics analyses with these, but again, we'll move into systems biology analyses and other methods to analyze them, again, having to think outside the box because we will be integrating all of the data together, and that this deep clinical phenotyping and longitudinal follow-up will actually help to understand some of the relationship between the clinical parameters and the panomics profiling and outcomes. And I will say that on Sunday at ATS, the first data from PVD-omics is going to be presented. So there's information coming from the first 600 participants. Now, once we have the data set and as we get farther along, the plan is to do what many people have shown here before, and that is to use the data for a discovery set, split it in half to have a validation set, and then again, work with many people in other cohorts to cross-validate to ensure that what we're seeing here is not unique to the cohort that's been recruited to the study. And as always, this study could not be done without the large group of people that are here on this slide that have all contributed, and this group and many more who are behind the scenes. So I want to say thank you for the opportunity, and if you're interested in more, Sunday afternoon, come see the data sets presented. Thank you.
I did have one quick one through the internet. In patients who participate in PVD-omics, is there a plan to collect lung tissue at time of death or transplantation?
That's an excellent question, actually. That has not been part of the original protocol, and I think we have not had any participants who have been transplanted yet. So, but I think, very good point.
Absolutely. Brigham, did you have a question?
That was my question. The spirit of it is, the spirit of that was to see, as you said, the strength of this is the approach that you have taken to collect blood from multiple different places or samples. The question becomes, does that reflect the pathobiology that's occurring in the lung? And the longitudinal aspect of that is a little bit confounding, but it would be great to have that tissue at the end. To make those kind of observations. So
I will say one thing towards that side. As one part that's not part of the main protocol, there are groups collecting endothelial cells using some different methodologies, and some of that data should be forthcoming.
It seems like a tremendous effort, and I'm sure it's taken a huge amount of time and energy to get
It's a group effort. This cannot be done without the input of a large number of people.
Yeah. How do you anticipate, with this breadth and depth of data, how do you anticipate putting it all together? Is it going to be a completely unsupervised approach, like Andy's approach, sort of see what maps out, or are you going to go looking to answer specific questions?
No, so much like what Andy was talking about, we favor not going with a reductionist approach and not focusing on a single pathway, but to really have a little bit more of the true systems biology approach where we're there for discovery. So I completely agree with what he had said in his approach as well.
Jane, I think this is the first time I'd caught it in one of your presentations that there's actually childhood data and exposure history being captured in these. What sort of information are you guys obtaining?
In terms of exposure history and female reproductive and things like that?
You said childhood data even.
Right, well, yes. So for all of the individuals, there are hundreds of questions that they answer on the survey. So it talks about childhood, history, medical history, and exposures from everything from molds, animals, altitude. It's a pretty extensive variable screen. If you're asking specifically about gestational age, we did not ask that in the female reproductive history.
We have, certainly my interest is in prematurity related outcomes, but we also have some survey data that's not published yet that would suggest early life respiratory infections being an increased risk factor. So these are things that we're not capturing in any of our sort of databases right now, which is unfortunate.
It's an excellent point. I think it's being missed. Yes.
Are the samples anonymized, or can you go back to the patients and do that sort of thing? Take additional samples, take additional history, or you can go back to patient records in hospital and
right. So
primary care.
We in every center can go back and get whatever we need. Because we know who our patients are. As they get coalesced into the study, they are being given codes and being anonymized. But again, if the centers have written it into their protocols, we can actually get longitudinal samples and also longitudinal histories, and even more importantly, right heart cath information, treatment information, and see how it all correlates over time.
I guess I'm just wondering whether you can physically go back, once you've got the data set at the end, whether you've broken the link between the data, like whether it be genomics or whatever, and the patient, or whether you can recall by genotype, see what they're doing in 10 years' time.
Right. So I think it is all completely anonymized at this point. And so at the data coordinating center, even once they break it, they will not have the information about the participants. They will have, the plan is though, they contact the site, and the site has the clinical information and they can go back. So it's to keep it, and it's done for privacy concerns and to keep everything separated so that the data coordinating center doesn't have PHI.
Great. One last question.
Fantastic data set. How do we know that the plasma signature we're seeing is reflective of pulmonary pathobiology and not right heart pathobiology? And is it important at all?
Right, I think we can only figure out correlations and associations at this point. And we have extensive RV phenotyping being done by echocardiogram, cardiac MRI. And it can, could potentially be from that. As sub-studies in some of the centers, they are looking at patients who do have RV dysfunction and who do not have RV dysfunction. And so this too is coming up as a potential way to look at it. And there's even, again, not linked to this study, but with some of the same cohort, people looking at PET and the vasculature. So you can see whether you've got vascular remodeling independent of RV dysfunction. And that really will probably be the only way to truly nail that question.
Fantastic. Thank you, Dr. Leopold.